Our next speaker is Aravind Asokan. He's a professor at Duke University. His work is really prolific, from the basic science of how AAV works in the body to new payload strategies, and he's also an entrepreneur. Take it away, Aravind.
Thanks for having me. So my lab, I'm an organic chemist by training, and then somewhere along the way switched to virology. And these are the folks that do the real work. I'm just here to talk about it. My lab has been focused on multiple platforms, broadly looking to improve gene therapy. And we cut our teeth on a lot of AAV biology, which then evolved into engineering viruses. We've moved into some protein engineering and also some RNA biology that we've been tapping into off late. There are three sort of pillars here. I just wanted to highlight a couple of those. We're digging deeper into non-mammalian AAV because I think there's a lot of biology in there that's left to be understood. Also, the elephant in the room is there are folks with pre-existing antibodies, and I think there are multiple groups and entities trying to develop strategies to get around that. Well, a lot of these viruses are about 50% similar, and I think regardless of how much engineering we're doing, getting to 50% is still a very non-trivial scenario. So we're trying to tap into that. There's some biology there. I won't be talking about that in detail today. There's no need to rest everything on the capsid. We've been looking past that. These are some enzymes that we've been engineering to cleave IgM and IgG. They block complement really effectively, and this is data from non-human primates showing that you can rescue expression in animals that are seropositive. I think there are multiple platforms being developed in this space, and I think this continues to be an important topic. But what I really wanted to touch on today was this seemingly weird marriage between AAV, a DNA virus, and RNA. John provided a really clear perspective on this, and I'm just reiterating the point. This is a simple plot on ClinVar, where you have the size of the cDNA, and then you look at the number of mutations. This bucket on the left is all of the genes that would fit into AAV, and even those, some of the autosomal dominant ones that we just randomly highlighted here, are obviously not going to be correctable by gene supplementation. And then on the right side is outside of the packaging capacity, and you've got a lot of these along with some autosomal dominant ones as well. So how do we sort of continue to lean on AAV, where delivery clearly to organs such as the brain, muscle, and heart are still highly effective, but at the same time come up with modalities that are going to live within the means of what that affords. Let's see if this will move. Oh, there you go. All right, so a quick point I wanted to make was we actually got into the RNA space primarily with a very simple premise. Why not try to extend the half-life of the AAV transcript? And as I'm sure many of you are familiar, the circRNA space has taken off with a simple premise, again, that there's no ends, and so degradation is reduced and half-lives are increased. And I put this collection here, the many groups that have worked on these platforms, I put this collection here because the common denominator is all of these are Duke related, and I'll touch on some of these in a second. But with AAV, we really got into back-splicing introns, and that led to expression of circRNAs, which of course have a variety of functionalities that you can engineer onto them, whether it's sponges or splicing and so on and so forth. And what we did with that work was we actually developed simple back-splicing mechanisms. You can actually embed tRNA-based circRNA generation, where it just pinches off from the end of the tRNAs to make tiny RNAs, circles, and you can embed that within this intron because it's an independent splicing mechanism. You'll get both species of circRNAs, if you will, and we've now got non-human primate data, and this is all being moved forward at Torque Bio, which we spun off back in 2021. But what I really wanted to talk to you about today was more on theme with some of the points on genetic intervention and some of the points that John discussed, which is, can we just deliver a fragment of RNA that essentially invades the existing RNA and then tries to replace multiple kilobases, if that's a possibility? And it's just amazing to see. This was Bruce Sullinger's postdoc work with Tom Check and published back in 1994. And he says this at the end of his abstract: targeted transsplicing represents a general means of altering the sequence of specified transcripts, may provide a new approach to treatment of genetic diseases. About 5 years later, of course, this is with ribozymes. So he's looking at the tetrahymena self-splicing intron here. With this, I think we have other splicing-based mechanisms, spliceosome-related, that Mariana Garcia Blanco brought up while he was at Duke. And of course, that's been developed into multiple applications, and Ascidian and other companies are moving that forward. We developed, just to understand the biology some more, using a CRISPR-based approach where we used an inactivated form of Cas13 that essentially binds, and you attach the guide RNA to the rest of the transcript that you're trying to replace. So this is a simple GFP assay. So you've got a stop codon here, and then, of course, the exon on this side. And when this invades, enabled by dCas13, you're able to get improved transsplicing efficiency, probably simply because of the protein being able to clamp onto the RNA there and reduce the threshold needed. And that's on the 5' end, and that's on the 3' end. But this was really, the main purpose of this was we were trying to understand the rules of the game and engagement there. And of course, it also led us to think about how we would need platforms that we're going to continue to develop. So this is essentially a guide library, not unlike all the work being done in the CRISPR space and the siRNA space. And you sort of just walk the transsplicing constructs through your favorite intron. And you can, of course, pop that into a reporter system, or you can do that endogenously in a barcode setting as well. So just to give you an example, turns out a lot of the genes are related to heart and muscle. I think we all know that. And so we thought we should look at some of these and try to pick some. Titin, of course, is a monster, although it's a molecular biology playbook because of all the introns you can chase. So we went after something slightly less crazy, which was MOI87. And MOI87, as you know, encodes the beta-myosin heavy chain, and it's about a third of the HCM cases, lots of mutations, autosomal dominant inheritance. There's also dominant negative versions of the protein. So basically, throw everything at it. It's a really difficult target, and you can't use it for conventional gene therapy, of course. And when you sort of break it down, you've got this intron in the middle that we liked, and so you probably want to be able to replace the 3' or the 5', and that way you're getting multiple mutations covered through this approach. First thing we did was we picked what we thought was our favorite intron to throw in there. I'm showing you one of these. There are other introns we've looked at as well, and simply put it into a GFP-based system. And then we're trying to achieve either splicing in to correct this half of GFP or that half of GFP, but really about intron discovery. Just summarizing a few of our efforts in this, some of our data in this space. So again, one of the things that we learned was you can't really, guide position cannot be intuited from RNA binding alone. This is something that we went in with thinking it was going to be exactly that. For example, you can see here, this intron position, guide position, gets you more editing for the 5' end, whereas something that's working pretty well for the 3' doesn't work for the 5'. So it's not the landing spot of the guide alone. There's additional elements here. And we've been looking at RNA structure primarily to try to understand this, and that seems to play a bit of a role as well. The other lesson we learned was guide length does not inherently improve this. With the SMART platform, again, it was 100, 200, so on and so forth. For 5' and 3', the way we're looking at it is there are certain conditions that will work for certain introns, and it's not always the same. And then the most exciting part was, obviously, we're in trying to make this CRISPR-free and move away from CRISPR, not having to add an endogenous protein. We've been mining many RNA architectures, and we've developed a high-throughput approach to figure this out. And I'm just showing you a sample of these. But there are several RNA architectures that will recruit the spliceosomal machinery to that site, allowing transsplicing to happen effectively. And we've been running some CRISPR screens to understand what are the key proteins involved. So just to finish up, essentially what we've done is this is the endogenous MOI87 transcript. And what I'm showing you here is trying to go after a mutated exon 23 by just replacing 23 with all the others there. And if it's cis-spliced, you should get this. If it's trans-spliced, you should get this. And as we quantify this, we're starting to see that endogenous transcripts can also be edited this way, moving from our assay itself. This is in non-human primates right now, so we'll have some idea of the efficiency in vivo, and that's kind of where we are. So overall, I think it's a seemingly non-obvious interface where there's AAV biology and then there's RNA biology, and then I think there are some new tricks for this old dog here that we should be thinking about. Thank you again.